deposits and animal deposits. This slide shows some specimens of Treptosaurs from the Deucerai shale. This is the whole type of Treptosaurus deuceri. This is a pretty complete fragment cone of Treptosaurus deuceri from the trilobite or the Deucerai shale at the old 42 road cut. And then this is another species that's common in the Deucerai shale, Treptosaurus fosteri, which is a little bit narrower, less, more gradually expanding form. So Treptosaurus from the Deucerai shale consist of nearly complete shells with most of the body chamber and nearly all of the fragment cone preserved intact. These indicate that Treptosaurus was a moderate to rapidly expanding orthoconic longicone that was generally between probably about 8 inches to a foot and a half in length, including the body chamber. The body chamber was about a quarter of the total length of the shell, has a dorsomyarian peripract. This is the muscle scar that attaches the living part of the animal to the shell. And there's no evidence of a ventral hypognomic sinus. This is a groove in the ventral part of the body chamber that you see in modern day Nautilus that allows this tube, this hose that allows this thing to jet around in the water column to protrude and move around. So these orthoconic forms seem to have a somewhat different methodology of moving around in modern day Nautilus. Specimens are often encrusted with whole peripheral bryozoan mats. That is, the entire shell is completely covered with a very thin layer of bryozoans. And Carl Brett and Gordon Bear and myself put together a paper in the olden days, was it 1989, where we documented the presence of these whole peripheral incrustations of nautiloids in the Ordovician and the Devonian of New York. And it appears that these things grew on the nautiloid when it was alive, and the nautiloids were up off of the water column because they have a very uniform coating all around the outside of the shell. The neat thing is in the Deucerai shell, you rarely get the outside shell preserved. You get the inside part of the shell replaced by calcite along with the cephalopod. But in these ones with the bryozoans on the outside, you can peel off the bryozoans and you can see the actual outside part of the shell. And what you see is a very fine ornament of longitudinal lyri and transverse growth lines. But the really neat thing is you see these stripes, these color bandings on the shell. So on the dorsal part of the shell, you have these longitudinal thin stripes, color banding. And so these things were counter-colored. The dorsum had the stripes. The ventral lacked any kind of coloration. Which again is typical of nectonic organisms in marine environments. Okay, this is a slide showing the biggest and the littlest species of Treptosaurus from the Cincinnatian. This is the bigger guy. This thing gets to be, this is Treptosaurus cincinnatiensis. Gets to be about a little over a foot in length. This is the body chamber from Liberty, a specimen out of the Liberty Formation in Oxford. This is all bryozoan here. You can see this real fine whole peripheral coating of bryozoans on the outside of the body chamber here. This is the fragment cone. Again, this is a rapidly expanding form, at very large diameter compressed siphuncle segments. In contrast, this is the smallest species of Treptosaurus. This is Treptosaurus transversum out of the coat, and it has very narrow, gracile shell shape very characteristic external ornament of transverse fine lyre, which you can kind of see here and up in this specimen here. The siphuncle is the same type as what we see in Cincinnatiensis. It's just much narrower in diameter. So this is a much more slender, gracile form. Again, the really neat thing about the Deuceroy shale is the fact that you have complete specimens and that you have the internal features so beautifully preserved. Section specimens, this slide shows you Treptoceros deuceroi, a pretty complete specimen here. This is the body chamber, the fragment cone, and this is the apical end of the shell here. And this is, again, a relatively complete specimen of Treptoceros phosphori, which is a slightly more narrow form compared to Deuceroi. And the segments, again, are slightly different. They're narrower, and they are more ventrally located in Fosteri, more in the center here, and more compressed in shape in Deuceroi. So you can actually tell these things apart if you have well enough preserved specimens, which unfortunately you rarely get outside of the Deuceroi shale. So in these forms you have these really beautifully preserved internal features. I don't know if you can see in this slide here, these are cameral deposits right here. They weather off, they defoliate down here. And then inside the siphuncle, you can see these parietal deposits. The whole apical end of the shell, the siphuncle here is filled with these parietal deposits, both dorsally and ventrally. And again, these are the things that counterbalance these longitudinal shells. As the name of this genus suggests, Trepto meaning changing and Ceras meaning horn, the characteristic features of this genus is that the siphuncle changes its position and shape as the animal grows. And this causes some complications when you're trying to differentiate different species of Treptoceros here in the Cincinnati area. The siphuncle segments usually are about 1/4 of the diameter of the shell initially, and they're compressed, heart-shaped. As the animal grows, the siphuncle becomes less compressed and more depressed, more tubular, and it makes only about 1/8 of the diameter of the shell adorally. You can kind of see this in Treptoceros deuceroi. Here you have these compressed heart-shaped segments down here. By the time you're up here by the living chamber, you have an almost tubular-looking segment there. So we differentiate these things by their internal features, but in this form, the internal features change as the animal grows. The siphuncle changes its position as well. Initially, the siphuncle segments are mid-dorsal or central in position, and by the time you get to the dorsal end of a mature animal, the siphuncle on the ventral part of the shell. Numerous species of Treptoceros have been described from the Cincinnatian area. Unfortunately, most of these were based on fragmentary specimens representing different stages of the same of growth in the same species. So different parts of the same animal were often described separately as distinct species, because a lot of the times these guys were working with relatively incomplete fragmentary material. So it's very hard to differentiate Cincinnatian species of Treptoceros from one another and to determine which name, which you could have 5 names attached to the same critter, actually has priority. And what the tale is, that what the message from Treptoceros is you have to have complete specimens that are relatively well preserved to be able to differentiate nautiloids at the genus level, or at the species level, even maybe at the genus level. These things had to have a fine balance between, as they grew, they had to balance out the mass and the buoyancy of the shell as the animal changed its buoyancy or its mass as it grew. And so it was constantly having to readjust this mechanism to keep this shell at an even keel. And this is what we see in Treptoceros, and it's probably characteristic of a lot of other longiconic nautiloids as well. We just don't have